floor is yours. Thank you very much.
Thank you very much, and thanks, David, and to ask me to come down here to give this presentation. And I do the coding. I hope I'm not going to be eliminated very soon. So today I'm going to talk about the GenAI for tox, and as well as to support the drug safety assessment. And I'm not going to reiterate how important the NAMs and to help us to reduce the animal use. And Nicole already gave a wonderful presentation on that. I just want to point it out that in the end of the 2022, and the FDA's Modernization Act 2.0 was signed into the law by our president, still is the president. So, which is really energized the FDA to look closely on these NAMs for the regulatory application. And we even have a program called ISTAN, and ISTAN stands for Innovative Science, with science and technology approach for the new drugs. And that basically means any NAMs, including the AI, the machine learning method, and once being qualified by this program, you can use in the drug development, and you can send these data to the FDA without any further confirmation. So it's a tremendous incentive. So how the AI and the machine learning going to play a role in this equation? So I put on here just a generic framework of what we do, the toxicity assessment or risk assessment or whatever you call it. You have the compounds of interest, you put it in some sort of the testing systems, or the animal study, or NAMs, or the cell printing, and Paul gave a wonderful presentation on that. And then you generate a bunch of the data. And once you have the data, you look at the data, you make the toxicity assessment. Everybody doing that. So in the past 60 years, an AI and machine learning always play a role and to help a human to look at the data. We use the machine learning method to look at the patterns, which patterns will associate with some sort of the toxicity. QSAR is a very good example of it. So we're going to continually use this methodology. However, the most exciting part in the past 10 years is generative AI, right? When I say generative AI, you're probably first thinking about the ChatGPT. But GenAI and the predictive AI is the two branch of the statistics has been existing in parallel for many, many years. Only the GenAI was took off in the last 10 years. Of course, the ChatGPT is the successful examples to use in the natural language processing. And GenAI can use many, many ways. So the one I'm going to talk about is how we can use the GenAI to look at the study design and generate animal study data in the first place. So this is basically serve as a virtual animal model, as a digital twin for the toxicity assessment. So, and in the past several years now, and we developed GenAI models and as a virtual animal models. The one I'm going to talk about here today is the animal GAN. I'm going to explain to you a little bit more later on. And basically our rationale is we've been doing animal studies so many years. And think about it, how many animal data we have in the industry, in the academia setting, and a lot of them in the public domain. So we ask ourselves, can AI learn from the past animal study data in such a way we will be able to generate the animal study results for the new and untested compounds? So the methodology we use called the GAN, Generative Adversarial Networks, and just one branch of the GenAI method. There are many others you can use as well. We happen to use the GAN. So the GAN architecture actually is pretty simple, right? So let's just say you have a rat, it's using here, was treated by acetaminophen in the 28 days in high dose. You have this data, you sit in somewhere and called the real test results. And also we can implement the deep learning models called the generator to generate a synthetic data. And then we can compare the synthetic data against the real data with another deep learning algorithm called the discriminators, right? We go and then the discriminator is going to provide feedback to the generator for the improvement if they see the difference. So you go these loops again, many, many times until the discriminators will not be able to separate the real data from synthetic data. So now we are cooking, right? We have a model. Let's just say this is called animal GAN. Now you have a new compounds coming in and you say, I want to know if the rat was treated by this new compounds in the high dose in the 28 days, what the results going to look like. So the animal GAN going to spell out the results for you, right? So and we decided to test this hypothesis, and we used a data set from the large public database called TG Gates, and this is, I believe this is from the Japanese consortium efforts, and a lot of people use this data set. They spent about 10 years, over $50 million to generate this data set. Very rich data set. So we did not use all of them. We took the 138 compounds and for the 3 doses, lower dose, medium dose, and high dose, and for each dose group, and we have 5 rats. And these compounds were put into the 4 different testing schedules on rats. That's repeated those studies, 3 days, 7 days, 14 days, and 28 days. And 14 days means the rat was consecutively treated for 14 days. On the day 15, rat gets sacrificed, all the measurement has been done. So the measurement we use in this study is 2 sets. One is the clinical pathology measures, another one is the toxicogenomics data. We use this data to develop 2 separate GAN models, and one called the ToxGAN, another one called animal GAN. And they share a very similar GAN architecture. I will get into the GAN architecture later on. So the ToxGAN is dealing with the toxicogenomics data, and the animal GAN was dealing with the 38 clinical pathology measures. So those are the 38 of them. And 11 of them is hematological measures and 21 clinical chemistry measure. Top 7 relate to the hepatotoxicity. Next 7, and we used to assess the kidney toxicity. All right, so this is the real study design, and we have a little bit over 8,000 rats on the left side. We split into two parts, one for the training, and which was 6,442 rats associated with the 118 compounds. So we're using this to develop the animal GAN. And on the test side, we have the 28 independent compounds not used in the training set. And then we use the animal GAN. Once we have the animal GAN model developed, we feed the test set information into the animal GAN, ask the animal GAN to generate a synthetic results for these 28 compounds. And then we compare the real experiment data versus the synthetic data. So the concordance is about 98%, and it's very, very high. And then we did 3 extreme testing, and the first one, we make sure the 28 compounds is totally not similar to the 118 compounds. So that was the standard QSAR method, look at the applicability domain and all of these issues. So next one we did, we make sure the 28 compounds belong to the therapeutic category is not represented in the training set. So the last one testing is we make sure the 28 drugs was approved by FDA in the later years compared to these drugs that was approved by FDA in the early years. So give a little bit more perspective application of these models. All the results came back as very much the same answer. And the next one we said we need to test in the external validation set. Actually, we did not do it. At the very beginning, when we sent the paper to the Nature Communication, they said you need to do that. So we have to do it. So we took another larger database called the drug matrix, and in the bottom table you can see they are not identical in terms of the study design, but they are similar enough. So fortunately, there is 70 compounds was tested by both TG Gates and drug matrix. So we can use this as a baseline to understand the difference between 2 study design, what the variation looks like. So we found about 82% concordance between these TG Gates and the drug matrix. And then we applied the animal GAN, the still the same animal GAN on the 355 compounds, which was not tested in the TGAs, the only tested in the drug matrix. So we give a prediction on these for these chemicals, and then we compare results against the drug matrix results. That was about 83%. It's pretty in line what we understand about both data set. So now, if you still remember, these are 3 steps. Once you generate the data, now you look at the data, you need to do the toxicity assessment. So this is exactly what we did, and we decided to take one parameter at a time, ALT elevations for toxicity or for liver tox assessment, because we have the FDA guidance to told us how to do the assessment. So what the FDA guidance says, if ALT larger than 3 compared to the normal condition and consider intermediate hepatotoxicity, if the between 1 or 2 considers a minor liver tox. And we look at the experiment data and also look at the synthetic data from animal GAN. So agreement, one is 100%, another one is 83%. And the next one, we did 2 parameters, an ALT and ALP, which was determining the liver injury pattern. That is important in the therapeutic application. This dictated what kind of the therapeutic option you're supposed to use when a patient experienced acute liver failure. So if ALT divided by ALP is larger than 5, we indicate in the hepatocellular injury, and less than 2 consider is a cholestatic injury, somewhere in between consider is the mixed injury. You can see the highlighted number, the agreement between experiment data and synthetic data is very high. Now, as I said, the GAN has been around for quite a while, 10 years, and I think 2014 was the first introduced. There is a lot of the variations of the GAN has been introduced. We tested some of them. For example, like a big GAN, the third bullet point called a big GAN, and big GAN can dissolve the entire image and then reconstruct it into 3D dimension to do a lot of works. And semi-GAN, and we can incorporate the QSCR into the GAN architecture. So we did all of that. But what both animal GAN and tox GAN is many using the first one called a C GAN, called a condition GAN. We give a condition, what the results can come out. But another GAN approach is extremely exciting, called the cycle GAN. Now, if I tell you, and if I ask you to think about the horse, and then you close eye, I'm asking you to, what is the zebra going to look like? But in your mind, you were thinking, well, adding a little bit more strip, it becomes zebra. If I show you a photo took in the summer, and I say, you know, what is winter going to look like? You're starting to put the leaves on the trees and all of that, right? So then the cycle GAN, what it's trying to do is to translate the data from one domain to another domain. So what is that? That's the translational research. That's exactly what we do. So we decided to have another initiative called the Translate AI. And basically using the cycle GAN with a lot of the engineering allow us to translate one type of the data to another type of data. We might be able to translate the rat to the human, in vitro to in vivo. So, and several projects was going on, and I don't think I have a lot of the time to explain all of that. Let me just give you one example. Yeah, let me just give one example. It's the algorithm we developed called the TransTox. So on the left side, it still is the horse and the zebra. You can see, and the red one, the red and the horse, and using the generator to generate the zebra. And then we can compare the synthetic zebra versus the real zebra, see what it looks like. But at the same time, so we're using the zebra to generate the horse, and then we compare it again. So we get a very complicated, the line was going on to minimize the loss. And we take the entire architecture into the liver and the kidney. And we translated the liver to kidney and kidney to liver. And of course, the devil always in the detail. So the papers are very much accepted by the NPJ and digital medicine. And we try to use this approach to address a number of the questions, but I'm not going to go through all of these and questions, but two questions I think is very important. And also in our mind, in terms of the regulatory applications. The first question is, if I have a biomarker, has been around for many, many years now, and then I have a synthetic data coming in, and I apply this biomarker on that synthetic data to do the diagnosis or prognosis, is that the FDA is okay with that? So that's the one scenario. And the second scenario is, if you have the synthetic data generated by fancy AI, and using this synthetic data to generate the biomarkers, can these biomarkers can be used in the real world setting? So we did all of that. I'm not going to get into detail, but results is very promising. So, what are we going to do with all of these GAN models? You can do a lot of the tricky part to do these GAN models. So this is the one thing we wanted to do, and we are mainly focused on the liver and liver tox and the kidney tox. And let's just say you have chemicals. We can using the tox GAN generate the liver gene expression data. And once we have this gene expression data, we will be able to understand that the underlying mechanism of the liver toxicity, right? So that would helps. And then we can using the animal GAN to generate the ALT, ALP, AST, and all of these important enzymes to see how the elevation looks like, and including like a total bilirubin and so on and so forth. So you add another layer of the information in the risk assessment. And lastly, we have another Gen AI method. I do not have time to talk about, or the quantitative knowledge and activity relationships. Till today, everything we do is based on the chemical structure determine the toxicology, right? We know that's not correct. And not the chemical structure to determine the toxicity. We have a lot of the information about the chemicals should be used to develop the models. This is the Q card score. So we can using these three levels of the information for the liver toxicity assessment. And then we can use the transtox, translate the entire liver profile into the kidney. And we have a lot of the liver data out there, but we do not have a lot of the kidney data, and let alone other organ toxicity. So this is going to be very useful. Once we translate it there, we can do a lot of things. All right, so today I just mainly talk about the animal GAN models and briefly touch on the Translate AI, but this is a part of our larger program we have called the AI program for toxicology, or the AI for tox. You type in AI for tox in the Google, it will lead you to our website. And the first one called the BirdTox, and the BirdTox, and we started this project a long time ago. And that by then, bird models is the only game in town. Now it's ChatGPT, Cloudy, and all of these. So we call the BirdTox, basically use the larger language model to dealing with the FDA document. We have the safety AI. This is a traditional QSI type of the approach with a few twists, and mainly to address these endpoints are important in the review process. We have the pathology AI, and using the AI to dealing with the histopathology data. The BGAN, as I mentioned earlier on, was used in that approach. So, and many people already pointed out that we are living extremely exciting era. The AI methodology just improves so quickly. So I certainly believe our only limitation is our imaginations. So I'm very fortunate to have a group of the young people working for me, and they certainly not a lack of the imagination. And I also very fortunate to work with several pharmaceutical companies, AbbVie, BI, and the GSK. They all put animal GAN in their pipeline, starting to generate the data and provide feedback to us, and we can use that to develop animal GAN version 2. Thank you.